All right, here we go again. 17.5 is about RNA. RNA, which stands for ribonucleic acid, and I think we've already discussed this at the beginning of the lesson anyway, or beginning of the chapter. It's one of the nucleic acids. You basically have DNA and RNA. Well, DNA gets all the credit, doesn't it? DNA seems to be what gets all the credit because it stores our genetic information. And so, yeah, I guess it should. It's pretty important. DNA is awesome because it stores all the information. It's the master copy, but at some point we have to read that DNA and turn it into and interpret it so that we can make a protein. And so, how does that get done? So that the DNA stays intact and doesn't get ruined, and so that it can be interpreted. Well, it's RNA that does that, and so because of that, because it involves a lot, you actually need three types of RNA. And so we're going to go over three types of RNA that are important in being able to look at the DNA, make a photocopy of it in a different type of way than we just looked at. When we just had a lesson on DNA replication, this is different than that. RNA is going to still read the DNA and produce a new molecule of RNA, and then it's going to read that RNA by another type of RNA that brings amino acids and makes a polypeptide. And there's even a type of RNA that says, hey, I'm just going to be here to make sure that this works right. And so there's three very important types of RNA that we're going to discuss that will help us go from DNA to a protein. Okay, so DNA is like the master copy, and we basically leave it alone. And so follow with me here in the PowerPoint. We're just going to scroll up. This is actually kind of a short lesson too, by the way, guys. So just hang with me here. The first important points that we want to make are these three main differences between DNA and RNA. The first one is that the sugar is ribose, and that's implied by the name. So deoxyribose nucleic acid and ribose nucleic acid. So obviously RNA has ribose instead of deoxyribose. If you forgot what that looks like, scroll back up to the beginning of the chapter when we looked at different nucleotides. And basically the sugar in ribose is going to have an OH group on the second carbon of the sugar. Remember, carbon 1, 2, 3, 4, 5. Carbon 1, 2, 3, 4, 5. On the second carbon here for this sugar, there's no OH group. So that would be deoxy. It's missing the oxygen. But on this sugar, you have OH there. So this would be ribose. So deoxyribose sugar and a ribose sugar. So RNA is going to have this one, ribose. It's also, while we're here, might as well point out this point. The second point is that it has uracil instead of thymine. Well, what is that? It's this nitrogenous base here. Well, that may not be specifically this nitrogenous base, but it might actually. Let me scroll up here for a second. Yeah, uracil. Indeed. So this would be uracil. I'm not sure what this would be, but remember, the nitrogenous bases, you have four different main types of nitrogenous bases, A, T, C, and G. But on RNA, you have a different type of nitrogenous base. You have an extra one called uracil, but that is not extra because it really just replaces thymine, which I think is this one here. Oh yeah, perfect example here. So this would be thymine. And you'll find thymine on DNA where there's like deoxyribose. But if it's RNA, instead of thymine, you'll have uracil. So it looks slightly different. So again, these are the nitrogenous bases, and this is important. A pairs with T, and G pairs with C. So what about this U? Well, since it replaces thymine, it does the same thing thymine did, which remember paired with A. So if thymine paired with A, then uracil pairs with A. Uracil pairs with adenine. So basically, thymine and uracil are very similar, except for two ketones, NH, a methyl group here. Uracil is missing this methyl group that thymine has. Cool. All right. So if those are the first two differences, what's the third difference? The third difference is that RNA is single-stranded. Unlike DNA, which is always found as a double-stranded double helix, RNA is just a single strand. It doesn't even base pair with another strand. Now, something important to make note of, in which we will go into greater depths down here, is that RNA, can base pair with itself. Isn't that kind of weird? So, like, if these bases here can base pair with these bases, then you'll have hydrogen bonds there, hydrogen bonding between the complementary base pairs, and so that'll actually give this single strand. It'll actually give this single strand of nucleic acid some structure. Okay, it'll actually give it some structure. So that's kind of interesting. But in general, you have a 3' end and a 5' end of the nucleic acid. So this would be the 3' end, and this would be the 5' end. So it's looped all the way around like this. All right. Some other differences are just to note could be that RNA molecules are usually much smaller than DNA molecules. So, for example, if a DNA molecule went all the way from the left of the screen here to the right of the screen, this was an entire DNA molecule. Basically, there's only going to be sections of the DNA molecule that code for certain proteins. Like maybe this section here will code for the gene for hair color. Well, there's going to be some RNA, a segment of RNA that's made that looks real similar to this DNA section. The DNA section that we call a gene that codes for a particular trait, we're going to have an mRNA molecule that will also code for that trait. And so it's going to be much smaller than DNA. Also, since DNA is preserved, we keep it inside the nucleus, and the rest of the RNAs are usually found outside of the nucleus, except for messenger RNA. So I should retract that statement. I'll just continue reading up here. The base pairing rules are still consistent. So cytosine still pairs with guanine, but this time again, adenine pairs with uracil because we don't have thymine. Uracil replaces thymine. And so now let's get into the 3 types of RNA, and then we'll be done with this lecture. We're going to go into much greater depth as to how these things do their job. We're basically just going to say what their job is for right now. And so that may seem a little, convoluted. You may be kind of still wondering or questioning how these guys do what they do. If that's the case, I'm going to make this video about transcription and the genetic code and how to translate the genetic code so that you can make the protein. I'm going to try to make all that real soon as well, like within this week, so that you can already start putting that stuff into context. Because just simply knowing what these guys do, these different RNA molecules, isn't very helpful until you kind of see how they do their job. But this is a rather short section just defining what types of RNA you have. But if you want to just stop there and let this week go by and maybe spend some time on your final project if you haven't already, and then start next week with transcription and translation, I don't think you'd get lost or get too far behind because we still have a few weeks. Sorry, I'm just taking this moment to, since it's a short video, to catch you up on the schedule. We basically have, let me look at my calendar. We basically have all of this week to finish this lesson. And then next week you could do transcription and translation. And then the next week after that you could look at mutations. And then you would just do your final exam. You would do your exam that last week too. So, but I wouldn't try to wait that long. I try to get it done as early as you can so you have time to work on your final project for me and your other classes and so on. So, all right. First type of RNA is ribosomal. The second type is messenger, and the third type is transfer. You can abbreviate those two, by the way, with using all caps for RNA and then using lowercase letters for the R for ribosomal. You see how it's lowercase r, RNA. and then it's lowercase m for messenger, all caps RNA, lowercase t for transfer. Let's see, where is it? Right here. Lowercase t for transfer, and then RNA in all caps. So you can abbreviate them like that. In fact, that's very standard to do that. So the ribosomal, or rRNA, its main job is to be the host. It's the host of the polypeptide making party. Because remember, our final goal is to make a protein, which a protein is nothing but a chain of amino acids, right? And so we're going to make this chain of amino acids. Well, where are we going to do that? We're going to do that right here at this ribosome. We're going to do that at the ribosome. So I'll show you pictures at the end too. Actually, let me just show you right now. Here's a ribosome. Here's a ribosome. Now it's actually made up of a large subunit at the top and a small subunit at the bottom, and they come together to form a ribosome. And another thing that's in the ribosome is proteins. So check it out. Here's a real ribosome. You have a large subunit and a small subunit, and you also have some ribosomal RNA inside of this. So it's basically made up of proteins and ribosomal RNA. So let's find a good. This one's good. You see how these are like those like ribbon diagrams for proteins. You basically have ribosomal RNA and proteins that make up the ribosome. This is just a general type of ribosome, large subunit and small unit. But the point is that that's the only place where ribosomal RNA is found, is inside the ribosome. But it's important to note that the ribosome also is composed of other proteins, which is kind of weird because all of this process is used to make proteins. So you can't make proteins unless you have a protein. How does that work? Like, who came first, the chicken or the egg? So that's an argument against evolution. But rRNA, ribosomal RNA, again, is going to provide the site where polypeptides are assembled. And again, all this will make more sense when you see it in action. But for now, just know that ribosomal RNA, or the ribosome, is the host of the party. It doesn't actually do any of the assembling of the polypeptide. It doesn't assemble the amino acids necessarily. Okay, the second type of RNA is messenger RNA. So mRNA is basically a transcript. It carries a message. Well, what kind of message does it carry? It carries the same message that the DNA had, because the DNA stays in the nucleus. It never leaves the nucleus, but yet protein synthesis occurs in the cytoplasm of the cell. So how do you do that if you can't get the DNA out and read it? Well, you don't. You leave the DNA in the nucleus, but you make a photocopy. And when you make a photocopy of the DNA, you basically are making a strand of messenger RNA. And so for each section, so like let's say you have a long section of DNA that goes from the left of my screen to the right of my screen. That's a whole section, this whole segment is DNA. Well, only a small segment of DNA will code for a specific trait, and that's called a gene, and that gene can be transcribed into a small section of messenger RNA, and that's called a transcript. And that transcript will leave the nucleus and go out into the cytoplasm to be translated into a protein at the ribosome and by transfer RNA. Transfer RNA is a super key player in this game. Transfer RNA is the one that does all the, if you ask me, all the hard work. It's the smallest type of RNA, but yet it's the one that carries the amino acid, and it's the one that recognizes the message in the messenger RNA. So you got all kinds of transfer RNAs floating around in the cytoplasm. Some of them, and they have amino acids attached at the top, and some of them are carrying glutamic acid. Let me show you what transfer RNA looks like so you can get a good image of this. I don't like the images in the book. They could have done a way better job. So, like, here you go. Look, this looks weird. Like, what are these blocks? Like, I get that they're trying to make it simple, but basically, a tRNA, the major components to a tRNA molecule are the amino acid that's attached at the top and the anticodon that corresponds to it, or that is basically that recognizes the codon on the messenger RNA. So here's your messenger RNA here. You got these codons, and the anticodons recognize the codons by using base pairing rules. And so the transfer RNA, again, the point I'm trying to make is that it recognizes and reads the messenger RNA and matches up to it, and then also carries amino acid. But this is not what tRNA looks like. This is, we already talked about this. It basically is one strand, 3' end to a 5' end, that loops around on itself. That's why the 5' end is back over here where the 3' end was. You basically have hydrogen bonding occurring here between these nucleotides, because remember, C pairs with G and G pairs with C. I don't know why they have G paired with U there. That's weird, and that should not be the case. I don't know if that's a typo or what. But another thing that's missing on this amino acid, it would be an uncharged amino acid, I guess. Here's a charged amino acid, or a charged tRNA, I should say. A charged tRNA is always going to have an amino acid on it. So here's an amino acid. In fact, let me find a better picture of an amino acid that actually shows the amino acid molecule. That one doesn't show the actual amino acid. Neither does that one. This one might do the job. Here we go. This is the one I was looking for, actually. So here's your tRNA molecule, but now it's missing kind of the nucleotides in the tRNA. But you see at the tip, you're going to have the amino acid, glutamic acid, and it's going to bond to the transfer RNA. Remember, the transfer RNA also has this anticodon on it as well. So the transfer RNA is super important because it's what's bringing the amino acids. So without going too much further into translation and protein synthesis, we're just going to stop there and just quickly point out some major regions on the transfer RNA, which we kind of already discussed. Down here at the bottom, you have what's called the anticodon, and it's basically a sequence of 3 nucleotides, and it's going to complementary base pair with 3 nucleotides on the mRNA molecule, which those are called codons. So the codons base pair with the anticodon. So that's an important region. The other important region is up here where the amino acid attaches, and that's called the acceptor stem. And it always has this sequence here. A, C, C, adenine, cytosine, cytosine. And actually it's always on this 3' end. So the 3' end of the nucleic acid has ACC always, and that's called the acceptor stem. That's where the amino acid is going to bond. I don't know why your book is pointing to this little purple dot here. That is not an amino acid. That's adenine. There should be an amino acid that goes there, though, is kind of what they're trying to say. Okay. We got to get into basically all this detail later, so I'm saving that. Here's a nice little table that summarizes the information from this lesson, that you have 3 types of RNA. You have their abbreviations if you want them, that we always use, and you basically have their function. The ribosome is just the site, is the host of the protein synthesis party, of the polypeptide producing party, and it's found in the ribosomes. So the ribosome, you have to remember, is made up of ribosomal RNA and proteins. The messenger RNA is going to carry the information from the DNA to the ribosomes. Okay, and what's called a transcript. The process is called transcription, and the molecule is called a transcript, an mRNA transcript. And then translation is the process by which transfer RNA will interpret the messenger RNA by bringing specific amino acids to the ribosome for protein synthesis to occur. All right. And so, yeah, the next lessons will be about transcription, how to make that transcript, from the DNA. The next lesson will be about genetic code, kind of how that works. And then the next lesson after that will be about translation. So how do we use genetic code to look at the mRNA and turn that into an amino acid? It's just a simple decoding system, that's it. But we'll also look at the kind of like how this happens in 3D, or we'll watch some animations too to really see it happen. If you don't remember, we actually did this outside as well with we did this outside with rope and straws. I don't know which class I did that with, if it was the current juniors or seniors that I have with, but I know we for a few classes have taken that outside and made little cardboards that had little like amino acids attached to the top. I try to make this real as easy to learn as possible because it's kind of an abstract process, and to be honest with you, getting kinesthetic like that and taking things out and tangibly touching them is a really easy way to learn this material. Doing little labs with little cutting out pieces of paper and taping it together. I know it seems elementary, but that's a great way to learn this stuff. So I'll actually have an assignment like that, I think, for you guys maybe, where you do that for a lab credit, because I don't think there's anything in your lab right now. So that's probably what I'll do. And then, but also the animations are just going to be really good and helpful for you to understand what's going on. When you really understand what's going on with transcription and translation in your mind, like you can see all the key players coming together, then whenever you read about it or hear me talk about it, it just is really easy, actually. So, all right. Well, that's going to be it for this lesson. Stay tuned for the next one. I'll try to get those to you ASAP so you can immediately start putting this information into context. Bye-bye.